Hi, and welcome along to another In Conservation With. I'm David Lindo, also known as the Urban Birder, and I am talking with Karen G. Lloyd. Now, you're going to see the interview, bam, straight after this because I had a problem when I first recorded it. So you're going to go straight into a presentation. But all I did previously, what you missed, is just talk to her and introduce what she did and how she got to be where she was. But that will be explained again during the course of the program. So I hope you enjoy it. Don't forget, please like and subscribe and tell all your friends about In Conservation With. And I'd like to thank also my good friends Kawa and Birds, Wildlife and Nature for sponsoring In Conservation With.
We hiked down with, I work with folks, Costa Rican scientists at Avsi the volcano monitoring and geohazards institute in Costa Rica. And so you can't actually go in the volcano, but we went in they have to go in the volcano because they have to monitor it. So I went down there with them. And this is us. I'm this one. You can see I like this view because it sort of gives you a feel for like the tourists looking at us being like, what in the world are they doing? And us with our hard hats on, because when you're going down in the volcano, the rocks can fall on you. They're not, they're not very organized. They're, just got thrown up by a volcano. Um, so it is a little bit daunting, some of the things we do, but we found life in that volcano. And this is the question that we're seeking to answer. This is me in the volcano, standing next to a fumarole. This is sort of what it looks like. This is one of my colleagues from Italy. We're always a very international group of people, and you can see the vapors, these fumes coming out of the earth. But these fumes are what are fueling this life, and so we wanted. I mean, it's our big question: can we discover new types of life on Earth? Which maybe it sounds like an impossible question, but it's shocking how much stuff is here that we didn't know about. Another place that I look for this are deep sea hydrothermal vents. So these are places, they're not volcanoes, they kind of look like volcanoes deep in the sea where these chemicals come back up. And this is where my chemistry training comes into play because this new life that we're finding is really just a bunch of big catalysts running around running chemical reactions. We dive in this submarine called the Alvin. That's the US's only scientific deep sea diving submersible. We just have one. And it is federally funded, and I hope that that continues. And it runs from this federally funded ship, Atlantis. This is a picture of me getting on board when I was a graduate student with my advisor. There are three of us, and we dive all day long, and we collect samples, and it is pretty awesome. Things are kind of tight inside. This is me. If you look at all the lights around me, it sort of looks like an old set of Star Trek or something. It's because all that stuff was originally made in the 60s and it works fine. So let's not let's not make it fancy. You don't want to put too much electronics in there anyway, because you don't want the power. You don't want to have a fire. So this is the kind of stuff we're sampling down at the bottom of the sea. We're sticking a temperature probe with the titanium arm into these mats. So these white stuff and the orange stuff here are visible mats of bacteria. So it's not entirely true. I hesitated a bit when you asked me what a microbe is, and I said, well, they're tiny single-celled organisms. Well, sometimes they get really big, and sometimes they take sulfur into their bodies and become white and visible, or this pigment, this orange pigment, and we still don't really understand what it's doing with that pigment. So actually, we can see quite a few of these strange microbes with our eyes. But we also find really weird, big life down there, animals down there. And this is sort of the view. We have to have divers, swimmers, check us out before we dive. So I'm inside in this picture, and there's divers checking us out to make sure that we're good to go. And then they go back to the big boat. We drop down and do our work. Um, but it's what I'm if you take one thing from this, just know that what we're discovering is changing our conception of what the range of life is on Earth. And if that sounds like maybe a bold statement, just know that it just is. We just, it's not just me, it's everyone in my field. We are discovering new stuff all the time. And now I'll show you a little bit of the nitty gritty that's not going to be very accessible, but just so you see like what the science look like. What does this look like? So it looks like this. This is an entry of one gene. This is a gene that we use to, it's like a name tag gene because it's pretty evolutionarily conserved. Um, this is a gene that I actually sequenced during my PhD, and this is in a public database. You can look at this yourself. This is completely publicly available and to share, but it's not, it's just like A's, G's, C's, and T's all put together. So what we have to do is we have to compare it to something like normal bacteria. So this is what E. coli would look like in the same format. And we have to compare this sequence to this sequence and see what they look like. So we make like a family tree of them. And when we do that, when we just take these raw sequences and we say, okay, who are really, really similar, and so they're siblings, and then who's really, really different, and oh, I don't know what you are, you're something really new. We end up with this big tree of life. And this is, we've had some adjustments since 1990 here and there, but this is the base tree. This is sort of where we thought we were in about, I'd say, the year 2000. This is what we thought we had. Three big branches of life. All of the diseases, strep throat, you know, a lot of, well, viruses are not on here, but any bacterial infection is all in the bacteria, cyanobacteria. Lots of hot spring bacteria that you see in Yellowstone are here. Archaea include things like methanogens that make methane. They're mostly happening in the background. They don't, for some reason, ever cause disease, and that is a giant mystery. We don't understand why the archaea, none of them have figured out how to cause disease. And then this is our branch on the tree of life. We're in the animals. So the things that we're finding were like this. They were like new deep branches on the tree of life. It turns out that the tree of life was much more like a bush than what we thought. You know, we're not finding new species. We're finding that we're switching the tree around. And it's really stunning how much new stuff there is. Yesterday, my student walked into her weekly meeting with me, and she plunked down the printout of what she's discovering in a hot spring in Chile. And she has 4 brand new phyla. So a phylum level is like all of invertebrates. And she just yesterday discovered 4 new ones. I mean, it's I can't overstate how much new and weird life there is inside Earth's crust. This is another volcano where I've worked. This is Fagradalsfjall volcano in Iceland. And so we went out here when it was erupting. Took this picture with my cell phone without a telephoto. It was an amazing volcano. But we were there looking around it in the surrounding area to see about how it's shifting the hot springs in the area and the life in the hot springs. So we were pulling out fresh lava. This is me banging on the lava. Oh, I think I'm, yeah. If you hear the tinkling, that's the lava cooling. It's the most amazing thing. And just sledgehammering freshly cooled hot lava for your colleagues is just, while it's raining in Iceland, it's just one of the best things in the world. I'm so lucky in this job that I get to do cool stuff like this. One of the microbes that people are studying is this one. This is Loki archaeota. It's now called Prometheus archaeota. And it's tiny. And this is probably the closest relative to us that's a single-celled prokaryotic organism. So this is the link between our branch on the tree of life, the eukaryotes, and all the rest of life on Earth. And we just discovered it in the 2000s. So this is, we're gonna see an explosion of understanding of where we are in the tree of life, definitely in the next few years. So, well, I'll go to the last place that I work, which is in the Arctic. I work 79 degrees north in Svalbard. It's a beautiful place, lots of glaciers, and we take this drill. We have to be armed at all times because the polar bears are everywhere. They are everywhere. So we do not shoot them. We actually do a lot of avoidance, but we have to be prepared just in case. This is our drill rig. I like this picture because this is me I'm talking with my colleague who's Russian, and this is my colleague who's British, but he works in France. And so I just want another big thing always want people to know is just how these efforts at finding new things on Earth have to be international, because if I'm spending a whole lot of time finding something that my colleague in France already knows, then it's a waste of everybody's time. So we have to work together. And I love the international aspect of it. It's been hard to work with my Russian colleague lately because our countries, it's hard to move between our countries because of the war. But I'm hopeful that we can restart that soon. So the two big ways this affects us directly are potentially with carbon capture and storage. This is one of the mitigation strategies for climate change, and almost all of the storage options involve some sort of sticking carbon deep underground or sticking carbon in the deep sea. And so currently I do not think that there's enough emphasis on the microbes that are already there and what they might be doing to that carbon when we put it down. So I think that this is an area that really could use a little more attention by we microbiologists need to be talking more with these industries to make sure that any sequestration projects that they do are effective. Another way that this directly affects us, or that we interact with these deep organisms, are that deep sea mining is an industry that is, I think, starting to ramp up. I can't tell if it's, a bunch of charlatans selling fake stuff, but they're definitely doing it. People are mining in the deep sea, and this is a big disruptor for deep sea life. It can technically be done safely, but I don't know if it can be done safely, if the amount of money you have to spend to do it safely would eat too much into profits and make the whole thing worthless. But this is something that I definitely am monitoring and keeping my eye on. I wrote this book. It was really fun. I meant it to be fun for people to read, hopefully. I did want people to know a lot about thermodynamics. So I've been told if you don't like the thermodynamics, you can kind of skip over chapters 6 and 7. But I like the idea of like the hard-hitting science to the people. I've heard from other folks that chapters 6 and 7 are very doable. They can handle it. So if you make it through chapter 6 and 7, please drop me a line and let me know that you enjoyed it, or if you skipped over them. Either way. But hopefully it's as fun for you to read as it was for me to write. I am funded by these people. Some are private institutions, but NASA, the Department of Energy, U.S. Department of Energy, and the National Science Foundation are federally funded agencies in the United States who have traditionally always been funded by the US government. And then I compete very hard against my colleagues to try to get these grants. It's very hard for me to get a grant, but the money is there and gettable. That's under debate right now about whether the US is going to continue to fund these agencies. And so I'm hopeful that the cuts have not happened, but I'm hopeful that the United States will choose to continue being a leader in science. And this is the way to do it, is to fund these institutions, because this is how we do blue skies research. No company is going to pay me to discover new life. There's no return on investment on that. And I'll leave you, or leave this presentation with this stunning photo of my graduate student, my former graduate student, Katie Sipes. This is in the field in the Arctic. We get all the way to the Arctic, and she goes up to me with her bag, and she's like, "Look at what I brought. I brought an Elsa dress." I was like, calling. So we took a picture of her. This is actually taken by Esther Horvath, who is an amazing professional photographer. She's a photographer for National Geographic, and so she did this portrait of Katie in the night. But anyway, I'm happy to chat.
Yeah, that's a beautiful end photograph, and I wondered how long that took to take because she couldn't have stood there for that long.
She was miserable, but she was like, "This is worth it."
Yeah, well, absolutely fantastic. Well, firstly, I really do hope that your federal funding continues because what you're doing, I think people don't realize maybe in the main, i.e., outside of your circles, but this sounds like an amazing sort of work. Very dangerous work. Talk about, chasing after lions and stuff like this is like very, very dangerous. I mean, you talked about polar bears. I mean, I was in Canada last month on the Northwest Passage, and when we got off anywhere, we had our bear guards, and sometimes we couldn't make landings because of the bears. And it makes you realize that if you ever shipwrecked in the Arctic and you're on your own, I'd be so scared if that was me.
Me too. It's there, they really are everywhere, and we do avoid them. I mean, it's their home. We don't, the only places that we shoo them away from is our little research village. Like if they come near the research village, we encourage them to leave. Like people will drive trucks over to them and be like, "Hey, go." Um,
They normally
but
take a hint, don't they? They normally take the hint, don't they?
they normally what?
They normally take the hint when you go and shoot
Yes.
off.
They are surprisingly recalcitrant. I mean, they are not used to having they don't have any predators of them. So they're not very impressed sometimes by our efforts to shoo them away. But if there is a polar bear anywhere around like where we're going out to work for the day, then we just can't go out. So
Yeah,
we don't shoo them away from our field site, just our living place.
yeah, fair enough.
We are actually very respectful of the bears, but we do carry a gun just if it comes to that.
You have to. And like you said, I love the internationality of all of this. It's just great for all. It just shows that the world can actually work together. I don't know what's going on with all the wars and stuff. We actually can all work together underneath it all.
Yeah.
It's just madness. But what struck me, especially having sort of looked at your book, is the fact that what I got from it, and I'm guessing a lot of people have got this as well, is the fact that you talk about or you think about oxygen and water being the classic necessities for life, and you think that all those things define life, and without them, there is no life. But you're saying actually there isn't that sort of scenario. It's actually some kind of energy that life depends on. Can you explain a bit about that?
Yeah, oxygen is just one of the specific things that all the life that we interact with needs, but it's not a requirement for life at all. When you get down to the nitty-gritty of what you really need, you have to have an energy gradient. So that would be like two molecules that are capable of reacting with each other. When they undergo a chemical reaction, they release energy. And so if you have that scenario where those two chemicals can interact and they can release energy, and you have a way to harness that energy and use it for things like breathing and living, running around, whatever you're turning your flagella, building a wall, whatever thing your cell needs to do. If you have that capability, then you can get energy from that. And so the more general way to look at the requirements for life is to look at it in terms of energetic gradients. And when you do that, it becomes much more varied. We're so oxygen focused, food and oxygen, we have to have those things. But you can replace that chemical equation with pure hydrogen and carbon dioxide. They're some of my favorite microbes take hydrogen, which would be the equivalent of our like candy bar, and CO2, which would be the equivalent of our oxygen, which is crazy to think about. Like carbon dioxide is our waste product, but they breathe it, and they can put those two things together and live off the energy that's made.
So all of this challenges our perception of what it is to be alive, really, and what is life. Excuse my ignorance here, but did life on Earth start from this kind of base in terms of single cell microbial stuff, or I don't understand. I mean, how did that work?
We do know for sure that life started as a microbial thing. That is definite. There's a lot we don't know about the origin of life, but it certainly we did not evolve to be multicellular until actually I don't know when. It's hard to pin a date on it because the fossils don't really start until things got calcified and appetite and bones and teeth make better fossils than soft tissues. But we have sponge spicules well back hundreds of millions of years ago at least. But Earth has been Earth for 4.5 billion years, and the earliest evidence for life on Earth is around 3.8 billion years. And so that life was certainly microbial.
So the microbial life now um, that you are discovering and you and your colleagues are discovering, I'm trying to understand because you showed the tree, the tree of life in your presentation, and you're saying that actually there's many more branches being discovered. It's become more of a hedge than it is a single.
Yeah. Like yesterday in my office. It's like constant. Sorry, didn't mean to interrupt you. Just to emphasize what you're saying.
Well, well, just as an aside, I think it's fantastic that you can discover something like that, like your assistant or colleague, or whatever,
Yeah.
coming back to you and
My
discovering.
PhD student.
Oh, that's brilliant. Are these? Are you actually describing these forms of life as individual species? Because I know you talked about bacteria which are harmful to us, like E. coli. And again, excuse my ignorance, but is are the E. coli bacteria, even though they're on the same kind of tree, are they kind of related to the other types of bacteria? And also, are all of them individually kind of classified as species, or how do you classify them? Because obviously, the other things we know are species. So how do you classify the microbial stuff?
We use species as well, and things get really hard trying to overlay species in animals and species in microbes because to be a species in animals, you need to have like separate mating. Like they can't like so there are a lot of different species in animals that could produce viable progeny, but they just don't because they never interact with each other. And that's so then they're different species. That that rule, we can't apply that in the microbial world because we don't even know where they all are. You can never really say for sure that something is not in a place because sometimes if you look harder, you'll find like one cell of it or something. Um, Plus, they mostly divide asexually anyway. I mean, they do undergo sexual reproduction as well. It's just very anyway. So for all those reasons, species, like as a word, we use that word, but it just doesn't map. So I think that's going to be an interesting area of taxonomy is people who are trying to merge the whole tree of life. And I know there are folks trying to do that. It's hard. But just sticking in the microbial world, we do have species. We have a official international committee for systematics and nomenclature. I always get the name wrong. I think it's something like that. And we're working with them right now to try to get them to name these uncultured, strange, deeply branching things, which currently they don't have a way to do that. So we are in a world where we kind of produce names for these things like whatever we feel like. We've been naming a lot of them after Norse gods lately. And then like everybody else gets in on the action. Like folks in China started discovering some of these like members of this group. So they named it some Chinese gods. And I know somebody who's Puerto Rican who discovered one of these groups as well. And so he's like, oh, I'm just gonna name it after a native Caribbean god. It's like great.
Have you got any named after you, Karen?
Well, I don't think I have anything named after me. And you
Why?
definitely
Come on, you should.
you,
I want a petition for this. I saw a
and
petition
you
to get
definitely
Karen a microbe.
you Karen a micro. You definitely can't name anything after yourself. That's for sure. I did get to name four. So we because the official nomenclature committee has, for the moment, I think they're going to change their minds. For the moment, decided that they won't be using this. We've had to make a separate organizing group called Seek Code, and so you can get official names through Seek Code. And I've named four of them, and I named them after a wonderful colleague of mine at Princeton who passed away, and I named one after a sort of unsung hero who's at North Carolina Central University. John Mayfield, and I just took a class from him for a summer when I was a kid, but he was very inspiring. So I named a microbe after him. He's a nice guy. He deserves it.
So, if there's all of this life that we don't know about on Earth, then what the hell is going on outside of Earth in the universe?
Yeah.
There must be,
There's gotta there's gotta be so much more, right? Like if it's this crazy here,
yeah,
gotta be even wilder out there.
yeah, because there's there's sulfur and all the stuff you talked about. That's all existing in
All
spades
all there.
elsewhere.
All you need are energy gradients and water. It does seem like you do need water, which is an interesting thought.
I saw you speaking, and you were talking about the number of bacteria in people's guts. If you added it all up, every single person in the world, it came up with an amazing figure in terms of tonnage of bacteria versus what actually is standing or living beneath our feet. Can you tell us again?
I don't remember the numbers because I had that in my second TED talk and I just don't I
It
don't
was
want
a
to
huge
the
amount.
numbers. It was a huge. It was, I mean, we're very focused on our gut microbiota, and we should be. I mean, I think that they play a really big role in our health and our wellness. But terms of tonnage, there's just not that many of them because if you think about, I mean, Earth is just gigantic. Um, it's hard for us to conceive of scale. I think so. I recently flew, I had a project in Guam leaving on a ship leaving from Guam. And so I flew from Los Angeles to Honolulu and then I flew from Honolulu to Guam. And in my head, they were just like little hops. But no, the Pacific Ocean is gigantic. And so I just was like, I kind of didn't pay attention to like how long the flights were because the landing and ending times are weird anyway because you cross the international date line. So I got on the plane and I was like, how long is this flight? And I was like, oh my God, 8 hours. Like,
Yeah,
what? It's
yeah. I
crazy.
I did a flight from Los Angeles to Honolulu once as well. It's a long ass flight. I mean, I couldn't
It
believe
is a
oh, it's a
long.
short hop like you. I didn't realize it was so long.
Yeah. It's so given that all the stuff in the ocean and all the stuff under the ocean for we don't know how deep it goes, at least 5 kilometers into the earth, every inch of that, every centimeter of that is packed full of living beings. And so that's how you can wrap your brain around those differences in tonnage. Like, 7 billion people on earth, all of the size of our colons. Oh, that's a gross image, but if you like took all our colons and piled them up, that would be like one part of the ocean. That wouldn't be the whole. We wouldn't cover, our colons couldn't cover all that space in Earth.
So I remember you saying that it was some phenomenal figure. I mean, it was a massive figure if we had all the humans bacteria, but to add or to actually just add up all the perceived numbers of bacteria on Earth, it was something like the weight of the Earth or something. It was just incredible. You talked about being underwater, but you've also gone underground, deep underground,
Yes.
as Jamiroquai once sang. You know that song? I'm going
Um,
deeper underground. Anyway,
no, but I like Jamiroquai. I'm going to look that song up.
look it
That's
up.
great.
Yeah.
Okay.
You went 3 kilometers underground in South Africa, didn't you? You
Sure
did.
did.
And you, by the way, guys, check out Karen Karen's Instagram because there's one of her
I
posts,
can put it in the chat if you want it.
one of her,
I,
one of her
oh,
stories involves her dancing 3 kilometers underground. Before we talk about what you were doing underground, surely that should be in the Guinness Book of Records for the deepest dance disco.
uh, deepest Tay Tay dance party.
Yeah, it has to be, isn't it?
I don't know. I don't know. That's not my field of work. I know more about the records of the microbes, but yeah, that was a spur of the moment decision just because we had to wait in that tunnel for there's like little trains that carry you all in between the tunnels 3 kilometers deep in this gold mine in South Africa. And so we had to wait for the train. So it wasn't disrupting any of the work. It didn't stand in the way of the science or anything. So I was like, let's just do the world's deepest
Listen,
take party.
Karen, scientists are allowed to have a bit of fun.
Yeah.
And
Well, but I do feel responsibility when I am in a submarine, when I'm, you know, I do take it seriously. There's a lot of resources go in to these sort of extraordinary things. And so I do try to not waste them. I'm conscious of that.
I know you don't, I know you don't. But when you were underground 3 kilometers down, I mean, firstly, before we talk about what you were looking for, what's it like being so far down underground in terms of, is it hot? In terms of, do you feel some kind of pressure, like in terms of the atmospheric pressure? And were you as scared as hell? You can answer in reverse if you wish.
Yeah. I was not scared as hell. It is hot as hell. It is, I mean, there's a reason that we depict hell as a hot place because it does like, and that's left over from heat of, are you hearing this helicopter? Sorry if you're hearing a helicopter. I don't know if Zoom is getting out of it. Um, the Los Angeles Police Department likes to keep a close eye on us, so they always fly over with helicopters. Um,
Can't hear it.
they're always around. We uh the heat of accretion. So when Earth formed, there was a lot of heat basically locked up in all these collisions, these massive violent collisions that led to the formation of our planet. And the planet has been cooling for 4.5 billion years. But that much heat takes a really long time to get gone. So we are still cooling down from the heat of all those accretions. And that's what makes the inside of Earth hot. So I was getting hot when I went down into this mine because of leftover heat from the primordial collisions that made Earth happen, which I think is cool to think about. Um,
It's mind blowing.
and yeah, and so I wasn't scared because I was just really impressed with the mining operation. I gotta say, it's just this is you know a commercially run mine. They have a ton of safety equipment,
What are
everything
they
with
mining? What exactly were they mining?
gold,
Gold. Okay.
gold. hmm but you have these giant cages, and that's how it's. They say that it's the longest elevator shaft in the world. It's a 2 kilometer elevator shaft, and then you get out at one floor, and then you walk over to the other elevator shaft, and then you go another kilometer down. And the elevator moves at 60 kilometers an hour, like a car. And so so you're standing in this room pressed against, so they push you in like cattle, and you're like there, it's the scientists, it's the miners, we're all wearing the same suits, we're all like decked out in our hard hats and our lights because zero light down there. And at one point my arm got stuck up and there was no way to get it back down again. So I'm like pressed against all these people with my arm up. And it wasn't scary because there's so many people and they do this every day. They go down there, and I was chatting with one of the miners, and I was like, well, what do you do? And she said, I'm a geologist. My job is to go around and check the stress that all these rocks are under so that we can stay ahead of problems and get structure support. And I was like, you have the coolest job in the world. And she was like, yeah, I know. I was like, this is so cool. But it was like meeting a famous person to go that deep in the earth. I had never done that before. It was cool.
So when you got down there, you did disco dancing. What did you actually find in terms of microbial life?
So I can almost never, so I just went there a couple weeks ago, and I can almost never answer that question this quickly after I get back, because the way we discover this life is it takes a lot of effort and it takes a lot of work in the laboratory. So basically now my students and postdocs are all haggling with each other to see who's going to get a shot at figuring out what life is there. So we're not quite there yet, but I can say what other people have found there. So I have a colleague, Devin Neeson, who's at NASA. She's working at NASA right now. She and her colleagues found more strange life. There is life in these brines. I have another colleague in Canada, Oliver War, who used isotopes to date. I don't know if those are all words that make sense, but he used chemistry to figure out how old this water is. And the water itself is over 1 billion years old. So this is water that has remained liquid for 1 billion years. Like, it was water, and these microbes that my other colleague was finding were probably there before any multicellular life, before sponges.
Did you take a sip?
Actually, I did accidentally get some because it comes out a ton of pressure. So it kind of sprayed everywhere. I was like, oh my God, I've got billion-year-old water all over me. And the thing I did not expect, I should have known this, but I didn't. It smells terrible.
Oh.
It was like putrid awfulness, just bleh, make you want to vomit. It was fantastic.
So you, in your book, you were talking about the fact that this microbial life eats or sort of sucks in the carbon dioxide, and it's a very important element that could help us in our lives on the surface.
Yeah. I mean, this is something that is being done, and to small scales and could be ramped up, is attempts to grab carbon out of usually point sources are a little more reasonable. So like a power plant, has sort of a lot of is producing a lot of CO2. If you take the CO2 instead of jetting it out to the atmosphere, instead you route it back down, um, put it into a liquid form, condense it down, put it under high pressure. You can basically just pump it down underground. And that is being done. And the thing is that these microbes, that's food for them. So they can use that and they can do things with it. And some things that they do with it might be good. Like if they turn it into rock, problem solved. And I want to be clear, like this isn't just like something we can do and continue extracting oil. This has to be combined with other attempts to change climate change, stopping emissions. But they could help, they could hurt. There are things that we could do with, if we mix something into what we put down there, it might, make things go a little better for the microbes. But we don't know. All that stuff is kind of in the future.
So kind of artificially feeding, I can't even call them wildlife, microbes.
They are wildlife.
Yeah, I guess they are.
Yeah.
Is that, I don't know, it kind of, I guess there needs to be more research done, but it sounds like, if you artificially kind of encourage something, it might have a negative side effect.
Yeah. I assume in birding, you would never put out, like, are you allowed to bait them? Like, can you put out? No.
It's not really. I mean, I guess it's almost akin to bird feeding, or feeding foxes, or feeding wild animals that hang out in your neighborhood because you
Right.
want to help them out. But that also has a detrimental, or can have a detrimental effect because it changes their behavior to become, habituated. Bears, for
Yeah.
example, become more aggressive or whatever, or more bold. So it's kind of interesting because even though they're single cell organisms, you never know what could actually occur from this. I mean, is there a case that we, like we have on land, on the surface, or in the seas, we need to actively involve ourselves in conservation in order to protect these animals and this wildlife? Do you think that could be a case for
I don't know.
microbial life?
Maybe. I don't know. We don't know enough about, we're just discovering them. We don't know how in danger they are from us. The only arena that I could think about for conserving them would be this deep sea mining stuff. That definitely will impact these deep sea communities. And hold on just a minute, I can let my dog out.
Okay, well, that's, once you get the dog out, it's really fascinating thinking about all of this because it's almost as if have you heard all of this, by the way? You've been hearing me now, yeah? You've heard.
Yeah, yeah, I can hear you just fine. I just had to, my dog was scratching at the door and driving me crazy.
I just, my head is spinning, and it's not often I do it in conservation with, and I'm left or even started kind of talking to someone, my head's already spinning because it's such a massive subject to get your head round. I've got all sorts of questions flying around my head. For example, how are these things born? What's day one look like? What does that look like?
Right.
You know?
What's day one? I mean, we know that they're born by dividing cells for the most part. So they go through mitosis, and we actually know a lot about how prokaryotes divide and grow. A lot of that stuff is targets for antibiotics. A lot of what we learn from it is because of diseases. Um, but we don't, we'll catch pictures of some of these cells in dividing states so we can see that they do that as well in the deep subsurface. But it's probably pretty different for them. I mean, the one really big, I think this is probably the most incredible thing about some of these communities is that they are so old and there's not a lot of energy for them. And so they probably haven't divided recently. So when we look at these microbes from these deep environments, some individual cells could have been alive for thousands of years or even hundreds of thousands of years, which is hard to even think about. Like, how do you slow down? How do you just not? cells are dividing all the time. Like the cells in our bodies are, we're constantly regenerating skin. We're like making new parts like all the time. How do you just stop all that and just sit for 100,000 years?
Is there a benefit to being so slow in your development, or is there nothing else to do?
So, right, that's kind of a debate that we're having right now is it a benefit or is it just, you know, they just forgot to die? Um, I think it has to be a benefit. I think that there's enough different groups that are doing this, and it seems to be their primary mode on Earth. So it seems like they are positioned to purposefully not die for that long. And so I always think, when we know about dormancy over shorter time periods, so dormancy over winter is something we know a lot about. We know that what they're waiting for is summer. Why would you be dormant for 100,000 years? Well, maybe you're waiting for the ice age to end, or maybe you're waiting for an ice age to start. And so they could be reacting to long, long time scale things that we wouldn't think of an animal ever reacting to.
What, I know you probably can't answer this question, but what do you think they are evolving into? Or are they evolving? Or is this evolution so slow paced that it makes our evolution from, whatever we were to crawling out of the sea to being whatever we are now seem as if it's the blink of an eye?
Yeah, I don't know. I mean, I think they probably do evolve more slowly because they do these long periods of not growth in their cycle. So that would mean that you would evolve more slowly as well. But they've had a lot more time to evolve. And you also have to think about like when you think about evolution, like, the difference between us and a bird feels, whoa, that's really different, right? Like really, really different. But like we both breathe oxygen. Like I can take a sequence of the cytochrome oxidases that humans use and a sequence of the cytochrome oxidase that a bird uses. And I can line up those two pieces of DNA. We can look at them side by side and see that, yeah, it's pretty much the same cytochrome oxidase in both of these species, even though we're totally different. We're talking about deeper branches on the tree of life. So things that have evolved away from each other so much that they're not even breathing the same things. They don't even have a cytochrome oxidase in some cases. So it's like, to answer that question, you have to be like, well, what do you mean by evolution? Do you mean like growing a new tail, or do you mean like figuring out how to breathe a rock?
Why, why, Karen? Why should anyone care about this life, this life form beneath our feet? Why? What? How does that affect? How do they affect our everyday life? For those
So
people
if,
who are actually obviously within your field, I'm talking about
right,
the vast
yeah.
majority of the planet.
I mean, if you think about what we discover when we discover new life forms, it's always something amazing. If we didn't realize that ants are so varied, they are, we now know they're incredibly diverse and they do so many things for the environment. And now we know that we know to conserve them, we know, you know, how to work with them better. Um, every time we discover the existence of a totally strange new group of life on Earth, it is a moment for opportunity. And we're doing that over and over and over again. And I can name specific things. One of the things that we do that we as humans use from novel life when we find it is that we learn from it and we do things. We can do things like it. For instance, there's a microbe that was in Yellowstone National Park that has an enzyme in it that is really good at making copies of DNA at high temperatures. And it's called Taq polymerase. And so we took that enzyme and we now use it. And when I say we, not just scientists in research, but medical people, anybody who's done 23andMe, it is the way that we make copies of DNA. Any work that we do with DNA for anything is going through this enzyme. We would not be able to do any of the stuff that we do with DNA if we hadn't borrowed this enzyme from this strange creature in Yellowstone. So when I think about the possibilities for discovery in this weird crustal life that we're looking, these deep sea weirdos, every single one of them could have an enzyme that's that impactful on human progress. We just have to look, and we have to look at them with that eye of like, how can I borrow what you spent 4.5 billion years evolving and use it? Use it for something good, make something cool out of it.
I wish I had a teacher like you. You are so enthusiastic and infectious, and I really feel like looking at the nearest uh, piece of life that I can't recognize straight away. I just feel very enthused. What is your favorite microbial life form? Have you one?
So I appreciate that you prepared me that you were going to ask that question of me, because my knee-jerk reaction is no, I have no favorites. I love them all equally. But I thought a little bit about it, and I realized I guess my favorite would have to be the Bathyarchaea, and that is a group. It is
Oh, what? Sorry.
mar- it's called the Bathyarchaea.
Okay.
Um, Bathy means deep. So like the bathysphere is the deepest part of one of the deepest part of the oceans. So my colleague Feng Ping Wong in Shanghai named it the Bathyarchaea, but my immediate colleagues when I was working in Denmark and I were the first people to get the genome of that organism. And I got interested in it because I was working in North Carolina for my PhD. And North Carolina is obviously where I grew up. It's not very exotic at all. And I was, just looking in the muck to see if I could find anything new. I was actually looking for a different set of microbes, but you get a list of everything that's there. And this organism was the most abundant type of microbe in this just normal mud that's like, by where my dad used to live. It's like not exotic at all, but this thing that was totally weird was in there, but all we had was one gene. So you just can't do a lot. You don't have an organism you can't see what its physiology is. Like, I don't know. So then for my postdoc, we were able to adopt a new technique for single cell sorting and whole genome amplification. And we were able to get an almost complete genome out of this organism. And that was really, when I got the annotations of what was in that genome, it was just a list of genes. I may have actually cried. I mean, I had been looking for this I've been looking for this microbe for years and just trying to figure out anything about it. And now I had a list of like all the genes it had. And so I could then go through that list and try to reconstruct what life it could lead based on the genes that I had for it.
What percentage of time do you spend crouched over a microscope?
Very little. Well, that's depends on the time of my life. I have spent like when you're doing a growth curve and you gotta count cells every few hours. Um, oh man, I've spent entire nights glued to a microscope. Um, it leaves like marks on your eyes. But I don't do a lot of microscopy right now. Maybe I'll. It just depends on what the project calls for.
If you could be anywhere in the world, where would you be now?
I don't know, right here because that's where my kids are.
Good answer. Well, I wish you all the best of luck with your book, Intraterrestrials, Discovering the Strangest Life on Earth. It's available from all good bookshops and a few rubbish ones as well, but you can definitely get it. And also, I'd like to thank everyone for watching this particular episode of In Conservation With. If you're watching this in the future on YouTube, please like and subscribe. I'm sorry I haven't got all the fancy whistles and bells and bells come up. I haven't got any of that stuff. But if you like and subscribe, it's filled with many conversations just like this, but covering so many different subjects. I think this has been the most fascinating. All of them are, but this has been really, I need to probably spend a week to sort of digest all of this stuff. But yeah, please like and subscribe. And at this point, I'd like to thank you very much, Karen, and wish you all the best.
Thank you. You as well. This is fun.
Thank you for being on this. And for everyone else, thank you for watching and keep looking up.